now we're going to move on to the development of the vasculature and your blood vessels. Now, what's interesting about this is that most of the organ development that we've talked about has been a single organ developing from sort of a single progenitor structure or precursor structure, or in some cases, two single structure, like the pancreas, which initially has two lobes that fuse into one. But the circulatory system, your vasculature, actually develops all over your body independently in many different places, and then as the vessels grow and develop, they ultimately merge to create this one large interconnected system. It's really quite fascinating how it happens. Okay, so just a couple basic terms that will help all of this make sense. And now we're shifting focus away from the vessels of the heart, although we will talk about them a little bit, but to the vessels that flow to the rest of the body. We have two types of blood vessels at a basic level. You have arteries. Now, an artery is defined as a vessel that carries blood away from the heart. And usually, that's going to be blood that contains oxygen, or oxygenated blood. Veins are defined as vessels that carry blood towards the heart, and usually that's going to be deoxygenated blood, so blood that's headed back to the heart to go up to the lungs to pick up more oxygen. However, there are exceptions. The pulmonary artery, carries deoxygenated blood to the lungs. It's an artery because it's leaving the heart to go to the lungs, The blood's flowing out of the heart, but it's deoxygenated blood because it's headed up to the lungs to pick up oxygen. And then we've got the pulmonary vein, which is sort of the opposite. So it carries oxygenated blood to the heart, but it's a vein because it's flowing into the heart. So those are the two exceptions to the rule. All right, so also obvious, I suspect, to all of you, blood vessels branch into progressively smaller vessels and then ultimately into capillaries. And it's the capillaries that sort of meet the tiny vessels in all of the organs that sort of meet and merge and allow for gas exchange and nutrient exchange. And it's at the level of the capillaries where blood passes sort of from the oxygenated system into the deoxygenated system. So a capillary is really neither an artery or a vein, but it's where we get flow of blood that's sort of technically oxygenated to become technically deoxygenated and head back toward the heart. Okay, so gas exchange occurs in capillaries, and there are lots of capillary beds all over the body. There are two different circuits that we refer to. The systemic circuit is the one that transports the oxygenated blood to the body, and then deoxygenated blood back to the heart. So this is basically everything except the lungs, upper body, lower body, everything except the lungs. And then we have the pulmonary circuit, which is really just the transport of deoxygenated blood into the lungs and oxygenated blood back to the heart. So two separate systems. Okay, how is it that we make blood vessels, wherever it is that we make them? Well, actually, interestingly, it begins with the yolk sac. So it begins in an extra embryonic tissue, a tissue that's not going on to be part of the baby. And this is also an interesting system because you get development of blood and development of blood vessels at the same time as part of the same process. So it's almost as if you were trying to plumb a house by first creating water and then building pipes around it, which is sort of a strange way to do it, at least for plumbing. Seems to work for blood vessels. So, in the mesoderm of the yolk sac, we get hemangioblast cells that are differentiating from the surrounding mesoderm, and they're going to aggregate into these clumps. Getting ahead of myself here. So they aggregate into clumps, and then they differentiate into what are called hematopoietic progenitor cells. Just sort of a term for this intermediate stage of development of the blood cells. From there, endothelial cells are going to start to aggregate around these clumps of hematopoietic progenitor cells, and this combination of endothelial cells and hematopoietic, I'm never going to get that right, progenitor cells, that group of these two cell types is called a blood island, and there are lots of these. Now, from there, the just never going to get it. The hematopoietic progenitor cells, from there they differentiate further into what are actual blood cells or primitive erythrocytes. So they're not exactly the same as the blood cells or the erythrocytes that you have in an adult human, but they perform a very similar function. And then the endothelial cells that are surrounding the blood islands will become the capillaries, and they're going to grow and connect with each other as capillaries do when they're developing. Okay. Now, interestingly, this only goes on for so long. By day 60, embryonic day 60, blood production in the yolk sac stops. Okay, and we switch over to a different system of blood production. So initially, the blood for the yolk sac, the placenta, the baby, all being produced by this process in the yolk sac. But then we switch, okay, and actually initially, the blood that's going, once we've switched over to fetal production of blood, it's initially actually the liver that drives most of the blood production. But blood cells are produced in the liver, the spleen, the thymus, and then ultimately the bone marrow. But initially it's the liver that's the primary source. By delivery, it's going to be the bone marrow that's the primary source of blood production. Now, when we're forming blood within the liver, there are two waves of hematopoietic stem cells that colonize the liver. So these are stem cells, so they're going to produce lots and lots of different blood types. They're not going to produce things other than blood cell derivatives. You're not going to make bones. So the first wave of cells, it's going to make a more primitive, they're more primitive types of hematopoietic stem cells. They're not going to be able to produce all the things that the second wave of hematopoietic stem cells is going to be able to produce. And it's those that really sort of get the circulatory system up and running. So they can make all the different hematopoietic stem cell derivatives, blood cells, immune cells, etc., including the definitive erythrocytes, which are the cells that are actually your sort of what you think of as a blood cell. They're what carry the hemoglobin, which in the hemoglobin is what binds to the oxygen, and that's what carries it around your body. Now, interestingly, not really an embryologic principle, but fetuses have different hemoglobin. So hemoglobin is that protein that sort of sits within your red blood cell that carries the oxygen, but fetal hemoglobin is a little bit different in its affinity for oxygen than adult hemoglobin. And people who have hemoglobinopathies, disorders where their hemoglobin is not functioning the way it should, like sickle cell, may actually, in some cases, continue to produce fetal hemoglobin, which is sort of a protective effect. And I guess outside the spectrum of this, or the purview of this course, but interesting, and certainly something that I hope you will learn more about in later courses, and I'm happy to chat about if folks have questions. Okay, so how do we make, back on topic, tangent done, how do we make blood vessels in the embryo, so in the tissues of the embryo as opposed to the extraembryonic tissues? In this case, this is a little bit more like plumbing a house the way that we think of. So instead of making the water and then building pipes around it, we're going to build pipes in one place and water in another place, and then have our water flow through our pre-made pipes. Okay? So there are two separate processes here. There's vasculogenesis and angiogenesis. There's just a subtle distinction between the two of them. In vasculogenesis, we're actually producing blood vessels directly from the mesoderm. So we're seeing differentiation of mesodermal cells to form blood vessels. Whereas in angiogenesis, we're seeing either sprouting or budding or division of existing blood vessels to produce more blood vessels. Okay, so in vasculogenesis, we see splanchnic mesoderm that produces angioblasts, which are sort of early blood cells, or early vessel cells, pardon me. And they do that because they're receiving signals from the associated endoderm. Okay, so the endoderm is signaling the mesoderm to produce angioblasts, which are then going to coalesce into vessels, and then as they grow and elongate, they will merge with other developing blood vessels and become the circulatory system. In angiogenesis, okay, again, we're starting with existing blood vessels, and either we're getting sprouts, like the branches of a tree, that grow off to form new blood vessels, or we're actually seeing division of a single vessel into creating multiple vessels. Okay, but both of these processes are at work within the embryo, in the embryonic tissues, and from this we get an entire circulatory system.